So here's a recap of what we talked about last week. That humans have essentially been selectively breeding plants and animals for the last 10,000 years. This process of domestication where we've said, oh, I like those traits, that has bigger seeds, that fruit tastes better. That's what I want when I plant these seeds again next year. And this domestication is this selection process that we've been talking about. Most of domestication, and I'll come back to this a couple times today, most of domestication is based on our phenotype. The traits that we can see, even though those traits are controlled at our DNA level, the specific genetic changes. But until the last 10 years, we didn't have the tools and the technology to be able to say, this change in the DNA is responsible for this physical trait. A taller plant, a plant that blooms earlier, a plant that produces more grain. So that's the background of what we've covered, of what we've talked about. We're going to focus this week on crop improvement. Next week we'll talk about livestock. So this week we're just going to focus on plants. So the big goals when you're trying to create better crops. Number one, lower inputs but higher outputs. So what does that mean? Less water, less nutrients. I can grow under soil that is less enriched soil. But for the output, I'm going to get more grain, bigger fruit, a higher yield. So if I can get more with less, that's the key. And then also, can I increase specific traits that really I find desirable? Maybe the way that something tastes. Maybe the color of the fruit or the color of the grain. How well it grows under challenging conditions. High heat, low amounts of water, frost, all those sorts of pieces. The hardiness of the plant. And then resistance. Am I resistant? Can I naturally resist pests? Can I naturally resist infectious agents, bacteria, viruses, fungi that normally attack the plant? Can the plant naturally resist that? And just a quick word here. Plants have evolved to resist the things that try to attack them. They produce substances that taste bitter so that animals don't try to forage from them. They actually produce their own version of pesticides. Most plants have naturally producing pesticides that are geared to kill many of the animals that generally try to attack them and suck out the sap. Many plants are naturally engineered, have naturally evolved to have resistance. This is the same thing we see when we talk about bacteria and the ways that they evolve resistance. When we look at things like the flu virus and how it continually mutates its DNA to try to better attack its host, better infect its host, and how the host the immune system is continually trying to change in order to resist or better attack back the virus. This same process is happening in all living things. Again, remember, one of our goals for this is to help us realize that this concept of natural is very different, that natural is in the eye of the beholder, because we have been modifying the genetic combinations in plants for 10,000 years. And we're going to get into some stuff today that is probably going to make you take a second look or rethink some of our definitions of natural. All right, now, this image, this is not in your notes, this image, these are pictures of this wonderful display that has been prepared by my team. They've done a fantastic job at the grocery store looking at all different kinds of fruits and beans and rice and grains and potatoes. I would encourage you to come and take a look at this at some point tonight. This is spectacular. There are probably 6 different varieties of peppers. And I know that there are 4 different varieties of potatoes. And I think there are 3 varieties of pears and multiple varieties of beans. And we've got wild rice and white rice. The crops that we eat have been domesticated to be as varied as possible, to be pleasing to the eye, to have different tastes, to have different sizes, to grow well under different conditions. This is not the way that these crops actually started. Most potatoes, actually, it's thought that potatoes originated probably in Peru, and the potatoes probably were skinnier than this, and they were all different shades of blue. but how often do you cook blue skinny potatoes at your house? You generally like the great big bakers, right? Or maybe the little red ones. So there's been an enormous amount of variety as our tastes have demanded things that look different or that are more exotic or that grow under certain conditions. And this has all been crafted by specifically breeding different varieties of plants. The tomatoes, we've got teeny tiny cherry tomatoes, and then we've got the slightly larger tomatoes that grow in the cluster, and then there are these enormous, as everything collides, these enormous beefsteak tomatoes. All tomatoes all originated from the same ancestor, but the specific genetic combinations that we've pulled together have given us all these different varieties. So again, I would encourage you to come up. You cannot take the produce home. And looking at all this produce makes me want next week to have like a giant like kitchen and stove and actually start chopping and cooking and doing those sorts of things. But we will refrain from being in Julia Child's kitchen for this week. And we see the same level of variation when we look at the floral industry. So this is just a beautiful image of all the different colored mums that are available in the fall. And what I didn't show you here is they're all different size mums. The big pom-pom mums that we wear on Friday football games, the spider mums that have the petals that branch out in all sizes, the little pincushion mums. Again, these all come from one plant that breeders have said, I like these traits, I like those traits. I'm going to breed and get those different traits. Again, I said this last week. This slide is common to last week. Historically, we've chosen traits based on what we could see, the phenotype, the visible trait, even though that's being driven primarily by genetics. Now we talked about that the environment plays a big role as well. Certainly the amount of water that a plant gets is going to drive how well it grows and what its flowers look like to some extent. But the majority of these traits are controlled by our genetics. And until recently, we haven't had any way to track or follow those. All right, we're going to spend a lot of time on the next two slides, and then I'm going to stop and ask for questions. Let's talk traditionally about how new breeds were made, how they were chosen, how they were domesticated or crossed. There's going to be a lot of vocabulary in this, so hang with me. So I have a tomato example, and this is a tomato that is an elite variety. So it's what you could buy commercially. Maybe if you're a commercial farmer, or maybe you have the Parks seed catalog and you want a certain kind of tomato, you could buy this. And you can tell this is a large tomato plant. It's got beautiful red fruit, big fruit, lots of fruit, but you can see these little flies around it. It's susceptible to a certain kind of pest. And maybe that's a pest that eats away at the leaves. Maybe that's an insect that bores into the fruit and lays its egg. But this plant does not have resistance to that pest. It's susceptible. So if I'm a seed breeder, I love everything about this plant except for the fact that the pests chew it up. So I'm going to try to create a variety that has all these great qualities, but is also resistant to the pest. So I look through all the different tomatoes that are out there, that are in my catalog. This is sometimes called my germ plasm. This is all the genetics, all of the plants that I could potentially pull into a breeding scheme. And I actually find this donor over here, and this donor is resistant to pests, no flies around it, but it has a whole lot of really poor characteristics. It's a small plant. The fruit is yellow and I want red. The fruit is small. So there's a lot of things I don't like about this plant, but the one thing I do like is that it is resistant to pests. So my goal as a breeder is to produce a plant that contains all the desirable traits of the elite variety, plus the resistance to pests, without any of the things that I don't want in my donor. Everybody with me? That's our goal. So here's how we're going to do it. We're going to cross our elite variety with our donor. So I'm going to take some pollen from my elite variety, and I'm going to brush that pollen onto part of the flowers from my donor, and I'm going to end up with a tomato that's got seeds, and I'm going to take those seeds and I'm going to plant them. So essentially I have crossed characteristics between my elite and my donor. And I'm going to end up with a lot of different plants. Some that are yellow, some that are red, some that resist pests, some that don't, some that are tall, some that are short. And I'm going to pick a set of plants that is closest to what I want. And remember, the thing I am always going to be selecting for is that they resist the pests. So right off the bat, I'm going to say, okay, show me all the plants that resist the pests, and let me pick one that at least looks kind of close to what I ultimately want to get to. So we're going to pick this one, which is red. That's about it. It's small, the plant is small, the fruit is small, but it's red and it resists pests. So now I'm going to take this plant and I'm going to involve it in a crossing, and I'm going to actually cross it to the exact same kind of plant as its parent. I'm going to cross it back to the elite variety. This is called a back cross. I'm crossing it back to what one of its parents was. And so I'm going to do that. And now I'm going to get a whole bunch of plants, and I'm going to select plants that, again, are resistant to pests and are even closer to the variety to what I'm trying to get. So in this case now, it's a taller plant. The fruit's still no bigger, and I still don't have any pests. It's resistant to pests. So I'm going to repeat that process. I'm going to do another back cross. Now I may have to do lots and lots and lots of back crosses. I'm showing you 3, but there may be many, many back crosses. And each time I'm getting closer and closer to the elite variety, but I've now brought in the resistance to pests from my donor. So at the end of the day, my goal is that I have a new variety that has all the elite traits plus the pest resistance from this plant, but none of the things I don't want from this plant. Make sense? That's my goal. That's what we want to get to. So let's now take a look at this. Let's lift the cover and look into our cells, and let's assume that we could look at the genetics at what is actually happening as we've gone through this process of creating back crosses to get to our new variety. All right, so the tomato, I'm going to just, for simplicity's sake, say that the tomato has 6 chromosomes. And that there are 3 different chromosomes, a short, a medium, and a really long, and there are 2 of each of those, 2 tall, 2 short, 2 medium. And I'm also going to say that we know the location of the genes, remember the recipes that provide instructions on the tomato that we care about. Now this is actually all true, but the tomato has more than 6 chromosomes, and I just made these locations up. But the story holds. So we've essentially said that on this short chromosome, right in this region, there's a gene that controls the height of the plant. And remember, we talked last week about this concept of alleles, which are different flavors or versions of the gene that lead to different traits. So at this gene, I may have some alleles that lead to a short height and some alleles that lead to a tall height. Now in reality, it's much more complicated than that, but it works for this story. And then on this chromosome, right here's a gene that controls the color of the fruit, and over here's a gene that controls how much fruit I get on the plant, how big the fruit is, and if the plant is resistant to my pest. So those are the locations that we're going to be following. And here, just for simplicity's sake, I've shown the genetic information from my elite variety all in black, and the genetic alleles from my donor all in white, just to make it easy for us to follow. Now here's the key. I want this plant to have all of the traits, all of the black alleles, except right here at pest response, because this plant does not resist pests, and this plant does. So if I could look at the chromosomes of this plant, I would want them to be black everywhere except in this one spot where I want them to have this set of information. So as we go through this process of crossing and pulling genetic information from each parent together, you can see here I've got half from my elite and half from my donor. And I'm going to cross it again with the elite. And there's a process that we're not going to dig into called recombination, which is why I'm ending up with this checkerboard pattern of black and white. But essentially, the more I cross back to my elite parent, the greater the percentage of the elite's alleles, or the black sections of the chromosome, that show up in my plant. So that at the end, when I continue to cross it, I get a plant, let me step out of the way. I get a plant that has all of the elite information except here where it actually has the information from the donor, which is what makes it resistant. Now there's another step that I'm going to go through ultimately so that both of these copies of the chromosome, both genes, have the white information. We're not going to dig into that, but every time, my goal is to have a plant that looks exactly like my donor, exactly like my elite variety in physical traits and in all of its genetic information, except at the one trait that I have tried to introduce in. And in that one trait that I've tried to introduce in, it now has the trait from the donor, pest resistance, and it has the genetic information from the donor that makes it resistant. All right, that's a lot of content to take in. So let me stop and let's ask for questions.
At some point, would you self-pollinate and then try to weed out the ones that to be sure you have a true breeding variety?
Yeah, that's a very good point. At some point, you're going to actually want to make sure that this plant breeds true. And that every time, this is what it breeds true. Now remember, we talked about corn last week, that corn is now available as hybrids, so it no longer breeds true. So that's a little more tricky for corn, but for a plant like tomato, if you were going to save the seeds, you would want a tomato to have the exact same, you would want the seeds to grow the same plant that the fruit came from.
So in a sense, you would weed out the undesirables.
You would be getting rid of the undesirables. Now, the thing that you can't control is you can't control that your neighbor's tomato plant might have pollinated, but the pollen from tomatoes generally doesn't travel that far. Yes, sir, and then we'll come up here.
You took the pollen from the elite and put it into the flower of the donor. Would it work if you went the other way?
Yeah, that's a very good question. I took, in this example, I used the words, I took the pollen from the elite and put it onto the flower where the egg is from the donor. Would it work the other way? Yes, it would. It would work either, in general, yes, I could have done it the other way as well. There are some plants where you might want to choose one primarily over the other, but theoretically, it should work in either direction.
When you're doing this, I assume this one up at the top has a good taste.
We're going to assume that this one right here has a good taste. Yes, we're going to assume that that was part of why it's an elite variety.
It's to have one with the same taste as the new one have.
Yeah. The goal is that this tastes, looks, smells, grows just like this one. Now, what you may discover is that in the process of this crossing, you actually got some little tiny bits of the donor elsewhere that actually might have even made it a better plant. It might have added a complexity to its taste, or it might have done some other things. But generally, if you have a best-selling variety, you want it to have the exact same stuff when you make the new variety, just plus that other new trait.
How long would it take for that process to go? It looks to me like it could go for several years.
How long would it take for that process to go? In part, that's going to depend on a couple things. One, how long do you have to have a plant grow before it's able to reproduce? So a tomato starts reproducing in a number of weeks. An apple tree takes a whole lot longer to get to the reproduction process. How many rounds, how many growing seasons can you get in a year? If I use indoor greenhouses, I can get multiple seasons of growing where I grow the tomatoes, I cross them, I get the seed, I plant the seed, I grow, I cross. You could get maybe 2 or 3 of those if I am able to be in a place where I can continually grow and plant and grow. If I'm in a climate where I only have a short growing season and then I have a really cold winter and I have to wait, and I can only do 1 generation a year, or I may have to wait several years like in an apple tree, it's going to take a much longer period of time. Yes, and then I'll come back to this side of the room.
I saw a special that some countries were going back to heirloom plants, and they were actually keeping heirloom plant seeds in a vault so that they could go back to them, and the countries were growing those better than they were the hybrids.
So this is the concept of a seed bank, that there are places around the world that store seeds from tens of thousands of different varieties. Number one, to preserve those varieties, and number two, in case there were some enormous disaster and we had to start agriculture from scratch, that we would have seeds. And many of them are the heirloom seeds, the plants that we or generations before us grew. There are some really unique things about heirloom plants. They have different growing styles, they have different flavor profiles, they are shaped sometimes more like tomatoes are shaped all bumpy and those sorts of things. The challenge, I love heirlooms, the challenge is heirlooms don't work so well when you're doing large-scale commercial growing because the plants might be producing fruit at different levels. That makes it very hard to commercially pick. The plants might flower over a month-long period, which means that when it's time to harvest, you may have to harvest multiple times and you run the risk of knocking not yet ripe fruit off. So there are some wonderful things about heirlooms. For large-scale commercial varieties, it becomes challenging.
Zeroing in generation after generation to that one point area in the final generation, how do you manage that? How do you know that area doesn't disappear early on?
Excellent. That's a great question. The question was, generation after generation, we're zeroing in, we're narrowing the amount of white that's on this map. How do we know that we haven't lost it? How do we know that it's not gone? Before genetics, before knowing the genome of the plant, the only way you knew you lost it is if the trait no longer showed up in the next generation. If all of my seeds now were attacked by pests, I would know that I had bred it out. If you'll hang with me for about 20 slides, we'll then talk about how the technology now lets us know that very, very precisely.
Can I guess you have to sequence each genome of each
You could sequence each genome of every seedling, but if you know the specific region you're looking for, so I'm cutting straight to the chase, don't get up and leave. If you know the region you're looking for, you can design a test that only looks at that specific site and very quickly says, does this plant have that site? Yes, no, no, yes, yes, and here are the ones I need to now work with. All right, let me see if we have any questions from our online audience. All right. I don't think we have any questions. All right, let me give you this example. Let me ask you this, because I'm noticing that my phone says that there are some issues with people being able to hear me. Will my IT crew, if you can hear me now, will you just send me a text and let me know that you actually can hear me, and I'll check that in just a second. All right, rice. This is a story of how a specific high-yielding variety of rice was created back in the 70s. I'm starting out with 6 varieties of rice across the top. Each of them has some trait that is useful. It produces high-quality grain, it's resistant to pests, it produces a lot of grain. The goal is to take all these parents and through a really complicated breeding scheme actually end up with a specific variety called IR36 that has all the traits that I want. This takes a long time and a pretty significant amount of money and a whole lot of rice to look at. But this is the process of bringing new varieties to market. It involves saying, I want this trait from this parent, and I want this trait from this parent, and this trait from this parent, and how do I go about adding one at a time, or stacking traits. I find this fascinating, but also really complicated. It makes my head hurt to look at this process. All is well. Good. All right. Now, here's the important part, and this comes back to exactly what you just said. The breeder has to wait until each generation of rice is fully grown to know if that rice has the qualities that they want. To know if that rice is high quality grain, how much grain it is, when did it flower, when did it mature, how long did it take. So you have to wait until you've reached that full maturity before you can say, yep, that's the plant that I want. Let's move it now through the breeding scheme. That takes an enormous amount of time. Some of the technologies we're going to talk about in a little bit really minimize that by knowing the genetics, by moving us from simply selecting on the phenotype, the physical characteristics, to being able to select on the genetic information that we know is responsible for that set of physical characteristics. Now, while we're talking about rice, let's talk about mochi. Anybody know what mochi is? Yeah, if you've been to Asia, if you've been to Japan especially, you know mochi. It's rice. It's a type of rice. Rice grains typically contain, rice is starch. They typically contain two types of starch, amylose and amylopectin. A single letter change, a single nucleotide change in one gene, and it's a gene called WAXY, W-A-X-Y. We have strange names for genes. One letter change in the WAXY gene means that that waxy recipe no longer can create a functional protein. And the protein that the waxy gene creates starts the pathway for making amylose. So if you have a mutation in the waxy gene, you get no amylose, which means that your rice only contains amylopectin. Amylopectin is sticky, really sticky. So the rice that has this mutation in the waxy gene is sticky rice. When you cook it, it does not separate. It's not the fine individual grains that you see in the commercials. It's like one giant mess of mushy up rice. And this rice is very, very popular in Asia. So it's often used, it's pounded and shaped, and sometimes it forms little packets that are sometimes filled with flavorings like red bean curd or fruit flavorings. And so it is a very popular rice cake that's often served around the time of the New Year, of the Japanese New Year. It also is found in these little teeny tiny bits that if you ever go to a yogurt store, a frozen yogurt store, and they have the bar that has all the chopped up fruits and the toppings and the chocolate and all those things, they also have these little tiny things that look almost like colored miniature marshmallows. It's mochi. So it is made of sticky rice. It's a little bit sweet, it's a little bit flavored. The different colors don't have different flavors. But that is caused by a single change in the waxy gene. One nucleotide, one chemical change is the difference between finely separated rice and sticky rice. When you in this room break tonight for cookies, there are a couple of bowls on the table that have mochi. They're little tiny pieces. Don't take a gigantic scoopful, but take a little mochi if you'd like to taste it, and think about the fact that it's made from glutinous rice that's a single nucleotide different from the rice that you buy in the grocery store that you might serve with, like, chicken. Those of you that are watching this at home, I apologize, I could not send you mochi. The break is probably not long enough for you to run out to the yogurt store and get some mochi, but the next time you're at the frozen yogurt store, take a look at that as well. All right, so we've talked about the importance of DNA variation. What causes it? What causes these changes that I keep talking about? The most common form of genetic change are errors in DNA replication. Every time your genome, every time your cells divide, they have to copy all the information in your DNA. And remember, DNA looks like a twisted ladder. We essentially unzip the ladder into two sides, and then we use each side as a template to fill in, to build back the other half. So every time your cells divide, we do this separation and this refilling, and it's called semi-conservative replication. Your cells have to copy correctly every single one of those DNA nucleotides, those DNA letters. And they do a really good job, but they're not perfect. And there's a whole set of repair mechanisms that come through and look and say, oh wait, that doesn't look right, let's fix that. But they're not perfect either. So for humans, on average, every time your cell divides, there's about 30 changes in your DNA that weren't present in the cell before. Changes that slipped through. Now remember, only about 1.5% of your DNA are the recipes. The rest is all this other information, this filler, for lack of a better word. It might be regulatory. So in all likelihood, those 30 changes are in that space. But if they're in genes, and they're in important genes, they potentially change the recipe. So that's where the majority of your change comes from. But another important cause for change are environmental factors. So this is things like ultraviolet light or radiation or certain chemicals that are called mutagens. They damage your DNA. And they damage your DNA by changing the chemical structure or by breaking the DNA, and it has to be repaired. And if it isn't repaired correctly, that's another way that you get mutation. So if we jump out of plants and into humans for just a brief second and we talk about cancer, we talk about cancer in Biotech 101 as an acquired genetic disease. In most cases, you don't inherit the mutations. There's a small number of cases like BRCA1 and BRCA2 and inherited breast cancer. But most forms of cancer, you acquire as your cells divide, they accumulate these kinds of mutations, either through errors in replication or through environmental factors. And at some point, if you accumulate enough mutations in enough critical growth genes, your cells go from a typical phenotype to a cancerous phenotype. Now let's jump back into plants. Mutations are a natural part of how we get our variety. They are what leads to all this variation. Changes in the DNA that got fixed, that didn't get fixed, that didn't get repaired, that then made it into the seed of the plant, and we ended up with a slightly different color or a slightly larger fruit or a slightly shorter plant. How likely is it that we would actually see a variant? So if I had a tomato plant and I wanted to see a change in a specific gene that changed the height of the plant, how likely am I just naturally, through errors in replication or through environmental errors, how likely am I going to find that change in that gene? Well, the typical mutation rates are really small, 1 times 10 to the minus 6 to 1 times 10 to the minus 8. I'm not very good with scientific notation and negative numbers. There is a 0 point and then a whole lot of zeros before you get to the number. So another way to think about it, roughly speaking, those of you that are math whizzes, hang with me. You'd need 1 million to 100 million seeds to find a mutation in the specific gene that you're looking for. That is a lot of work if you're a seed breeder. It's a lot of plants that you have to look through to try to find that if you're just going to allow the natural process of genetic change. But the natural process of genetic change is what got us to these plants, these different varieties. Mutations happen naturally all the time. And over 10,000 years of domestication and breeding, we found those changes that altered the plant a little bit. Now, here's what I want you to hear me all the way through before you jump up yelling and screaming. And remember our original rules. I won't yell at you and you won't yell at me, and I have fruit that I could throw. So, is there a way that we can increase the frequency of mutation? Can I accelerate that process? Can I make it so that it's more likely that I can find those changes that give that plant resistance or increase the yield or increase the nutrients? And the answer is yes, there is a way that I can increase the frequency of mutation. I can treat seeds, I can treat leaf cuttings, I can treat parts of the plant with chemicals that I know mutate DNA. Or I can expose those plants to radiation, X-rays or gamma radiation. And that exposure mutates the DNA. Now, it mutates the DNA at a pretty high rate. So that if I took 1,000 seeds and I exposed them to gamma radiation and then planted them, a whole lot of them aren't going to germinate because the DNA mutations did things that mean the plant's no longer viable, the seed is no longer viable. But some of those seeds are going to grow, and some of those seeds are going to have mutations specifically in the gene that I'm trying to target. Just like if it had naturally occurred from ultraviolet light, that type of radiation causing a mutation in the plant. And then I screen the next generation. Now, this is part of this joint FAO IAEA program, which is essentially something that was started around the time of the Second World War. It was atomic energy, nuclear energy for peace. It was a way to show that you could use the same kind of atomic energy, the nuclear energy that was in the bombs, that you could use it in peaceful ways by radiating seeds or leaf cuttings and inducing DNA mutations, and then screening to find those viable changes. This technology has been in use in our food for over 80 years. Because we have been trying to find ways to increase the frequency of mutations, and then looking for ways to screen the plant that's got the change that I want. Now the plants are, this type of radiation, the plants are not radioactive. Once you've irradiated it, that's it. There's no radioactivity coming back from the plants. And if you take that seed and you grow it, and then you take the seed from that plant, it doesn't continue to mutate. It isn't something that every generation there's additional mutation. It's a once and done kind of response, kind of reaction. So it primarily now uses gamma rays or X-rays. And you can also use chemicals. There's a certain chemical called ethyl methanosulfate, and that is a chemical that scientists work with often because it is very easily denatured. It's very easily broken down into harmless chemicals. So it isn't something that you have to worry about getting into the water supply and lasting for long periods of time. It is an easily disposable type of chemical, but it increases the frequency of mutations. And it increases the mutation rate 1,000 to 1,000,000 fold. So this is one of the machines that has that uses gamma rays. It uses cobalt-60, and you put the seeds in this dish and you close it up and it irradiates the seeds. There are places around the world like this where the irradiation is taking place in this concrete dome. There are fields that are spread around where you actually plant out the seeds and you look for your specific trait of interest. Now let me give you some examples of what this technology has actually been used for. Oh, let me back up before I do that. Let me tell you where it's used. More than half of the technology is used to produce grains. Grains that have higher yields, grains that are resistant, grains that flour faster, cereal grains. Grains like rice and wheat and barley are some of our major food sources. And in fact, Heather Smith, one of the members of our communication team, sent me an email earlier this week that said, let me get this right, let me read it, that said that 4 of the 30,000 edible plants, 4 of them provide 60% of the world's dietary intake. So 60% of our dietary intake around the world comes from 4 plants: rice, corn, wheat, and potato. So it's not surprising that grains, cereals, are at the top of this list. And then also flours, to get different types of flowering traits. Double blooms, blooms with color around the edge, blooms that have different shapes. Now if you look at 3,200 varieties have been produced by this technology for over 200 plant species, and most of them have been introduced in Asia, 60%, and another 30% in Europe. So a small percent have been produced in North America, and that was done primarily in the 50s and 60s and 70s, and then North America began to shift to more genetic technologies, more targeted genetic technologies. We'll hit that in week 4 when we get to genetically modified foods. A lot of you have sent me emails saying, are we going to talk about GM foods? Yes, we're going to talk about GM foods. Are we going to talk about big seed companies? Yes, we're going to talk about big seed companies, but not right now. I want to note something about this technology. This technology does not require labeling. There is no labeling from foods that are produced by what's called induced mutation. These foods also can be certified organic. So you have this very interesting situation where in Europe is very anti-genetically modified foods. Yet a large percentage, Europeans don't have an issue with food of this type and don't require it to be labeled. And this is, some would argue, a much more broad-scale technology where you're changing potentially several different genes at once than genetically modified foods where you're changing a single gene. Now, before I let you go for a break, let me give you some examples. Let's start over here on the right. Calrose 76 rice. This is a variety of rice that was produced in 1976 that is a higher yielding type of rice. It has now recently been surpassed by other varieties, but this was one of the primary rices that was used around the world for decades. And you can still buy big bags of Calrose 76 rice, produced by induced mutation, by irradiating grains of rice and then planting them and looking for the specific trait that you desired. Rio Star grapefruit. If you like ruby red grapefruit, I love ruby red grapefruit. Ruby red grapefruit was produced by induced mutation. We took grapefruit seeds from the pink-fleshed grapefruit, irradiated them, grew them, and looked specifically for plants that had this darker flesh. In addition to the darker colored flesh, these types of plants are they're cold hardier. They do better under cold conditions. So in Texas, there were several frosts that wiped out many of the other varieties of grapefruit, not these. So Rio Star, Rio, Ruby, Rio Princess, that entire group of ruby red grapefruit were originally induced mutation. Now, once you grew that first tree and you got the grapefruit from that, and you either grafted more trees or you took the seeds, however you did that, there isn't new mutation. This is no different than any other plant that had a naturally occurring mutation that I've now commercialized so that you can get the seed over and over and over. The seed breeds true. So it's only one time that you're trying to accumulate or trying to induce mutations, and then you've screened and selected for it, and then that plant becomes one of your primary commercial varieties. Golden Promise barley. This is a type of barley that was also produced by induced mutation, is grown a lot in Scotland, and is incorporated into many types of beers and many high-end whiskeys. In fact, there are a number of Scottish whiskeys that say on their label, we won't use anything but Golden Promise barley, because it gives us the flavor profile that is exactly what we want in a whiskey or in a type of beer. There are other examples of this as well. I just pulled out 3 that you're likely to run across potentially in your supermarket. And again, they can be organic, so you're likely to run across these in your Earth Fare or Whole Foods. And for 80 years, this has been a part of our system. And again, I'm not trying to say we need to, I'm not trying to drive us towards one technology is better than another, but it comes back to what's our definition of natural? How did we get these plants? How did we get these specific genetic changes and specific genetic variants? Okay, this is a great place for us to take a break. So let's take a 10-minute break, and then we will come back. Those of you that are here, don't forget to get a little mochi here. All right, let's move back in. We have a lot of ground to cover and not much time to do it. Wow. Okay, here are all of the plants that have had their genomes sequenced. So in other words, your genome is the collection of all your genetic information. And if you have sequenced the genome, then you now know what all the genetic recipes are in an organism. So there's a human genome. You've heard us talk a lot about the human genome. The first plant genome was sequenced in 2000. So we've sequenced all of these since 2000. Plants are complicated. They have lots of repetitive sequences of DNA. If you think about your genome as a cookbook, plants have lots of chapters repeated many, many times. They have lots of paragraphs from within a chapter repeated multiple times. There might be 15 copies of this paragraph on page 6, and another 4 copies of the same paragraph on page 35, and another 8 copies on page 76. So it becomes really challenging to try to interpret through. Lots of plants that have been sequenced, many of them that are important for food, but a number of them that also are really important for lumber, for example, or for raw materials, or for energy production. HudsonAlpha has a genome sequencing center. These are the people I told you about last week with the citrus. This group of people are amazing. They are brilliant at what they do. They work with scientists around the world and have built a reputation as knowing how to handle the challenges of plant genomes. And if you want really high quality work, you come to the people at HudsonAlpha. They're led by Jane Grimwood and Jeremy Smuts. They are amazing individuals, and we are really blessed to have them here at HudsonAlpha. They take us in an agricultural direction that our other faculty don't have. We hope to be able to expand that agricultural initiative. They do incredible work. I cannot say enough good things about this group. And this gives you a quick overview of some of the plants that they have worked with over the last couple of years. I want to point out soybean. They are the soybean experts. They really are. And I find it wonderful that every few years, the fields around HudsonAlpha, you know, they rotate what they grow. Sometimes it's cotton, sometimes it's soybean. And I love when soybean is grown. They've also worked on some varieties of cotton and the peach and the common bean and eucalyptus. Lots of different things. And I put this in here for you to realize, to see how many genes are in these different plants, and that there's a pretty wide variation, and how many millions of bases of A's, T's, C's, and G's are in the genomes as well. Lots of variety, lots of variation in plants from one species to the next. Now, so once you've got a sequence, so once we have the corn genome or the soybean genome, what do we do with it? Once I got a recipe, well, that's nice. I wrote a paper about it, so what? Well, that is just the beginning, because now you've got a reference you can compare back to. And now you've got something that tells you, oh, on this chromosome right here, here's a gene. And I think this gene is potentially involved in water usage in the leaves, or in the water uptake in the roots. Or I think this gene is involved in controlling when we flower and when we don't flower. So you can begin to compare varieties. For example, these are different varieties of Western Canadian wheat. Different sizes of grain, different shape of grain. And I can actually take each one of these plants that produce these different grains and go back and re-sequence them. So we've already done one sequence of the wheat genome. I can now sequence each of these different plants, these different wheat varieties, and I can compare it back to the reference, the wheat genome. And I'm going to find some changes between the reference genome and each of these varieties. And some of those differences in the genome are going to be due, or are going to result in, the differences in grain size. So it's a way for me to say, okay, well, let's take 70 soybeans that have different plant shape and different seed shape and different amounts of oil and different flowering intervals and different maturity times, and let me sequence all 70 of those different strains and then compare their A's, T's, C's, and G's with my reference. So I then build a catalog of where there are differences. And then I go through and I track those differences and try to figure out, what is this difference responsible for? Or is it responsible for anything? Is it silent? This difference seems to play a role in controlling grain size or in controlling time to maturity. And if I know those alleles, those variants, those changes, if I know what they code for, then I can use them in my breeding scheme to build, to crossbreed, to create a plant that has a set of characteristics that I want. Does that make sense? So the genome opens the door. Having the genome and then having the technologies that let you cheaply and quickly resequence or sequence through multiple varieties and identify where the changes are, that's where this power comes in. So let me give you some quick examples. Let's talk about corn. But first, let's talk about dirt. 50% of farmable land contains acidic soils. And one of the challenges with acidic soils is that the acid in the soil pulls aluminum out of the clay. So we end up with lots of free aluminum ions in our soil. And the problem with that is that aluminum damages corn roots. So if corn tries to take up the water with aluminum, the aluminum actually causes the roots to be stunted, to not grow well, and then the plant doesn't grow well. So here's a variety of corn that's resistant to the effects of aluminum. It's exactly the size you would expect. Here's the variety that's sensitive, so it's stunted, it doesn't grow as well. They're growing in the same soil. So the question is, what is it about the genetics, the genome of this variety of corn that lets it grow in aluminum soil where this one can't? So since the corn genome was sequenced, we can take each of these two plants, sequence their genome, compare them to each other, compare them to the reference, and try to identify what it is that lets this plant grow in aluminum-rich soil. So what we found is that there's a gene on chromosome 6 called MATE1, M-A-T-E-1. And this gene makes a protein. This gene is turned on to make that protein when aluminum is present and taken up by root cells. So when root cells take up aluminum, MATE1 is active. It makes a protein, also called MATE1. And that protein is it's a carrier protein. It carries citric acid from the root cells out into the soil. So these blue diamonds. So it transports citric acid from the root out into the soil, and that citric acid binds aluminum and prevents aluminum from being brought back into the plant. Really cool. Really a great defense mechanism. Smart plants. Most of the aluminum sensitive, well actually most strains of corn are sensitive to aluminum in the soil. And they have one copy of the MATE1 gene on chromosome 6 that came from the maternal parent, and one copy of MATE1 on the chromosome 6 that came from the paternal parent. The aluminum tolerant varieties have 3 copies of MATE1 on each chromosome, back to back to back. And so having 3 copies of MATE1 means you produce a whole lot more MATE1 protein, and you transport a whole lot more citric acid out into the soil, and you bind up more of the free aluminum. So by binding more of the aluminum, there's less available to be taken up to stunt the growth of your roots. So by comparing the genomes of the tolerant and the resistant, the tolerant and the sensitive strains, we identified this difference. This difference is called copy number variation. We see copy number variation in humans all the time. We have places in our DNA where we have large blocks of repeated DNA. Some of us have 3 of those blocks, some of us have 6, some of us have 1. This was the first time we'd ever seen it in plants. So the goal now is to figure out how to take this piece of DNA from this plant, from the aluminum tolerant plant, and cross it into those elite varieties that are commercially used for most varieties of corn. Another example with bread wheat. 1/3 of the world's population depends on bread wheat for survival, but bread wheats are always under attack from things like stem rust, which is a fungus. It's carried on the wind. The stem rust infects the plant, and it does a number of things, but one of the key things that it does is it disrupts the plant's flow of water up the stem. And so in a matter of 3 weeks, a completely healthy wheat crop is now reduced to something that is black, broken, and dried, and completely worthless. A few decades ago, we developed wheat rust-resistant varieties of wheat. Let me back up. We developed rust-resistant varieties of wheat, and that really staved off massive losses in the wheat crops. But there are new varieties of wheat rust that are mutating and evolving all the time, just like the flu virus. This is a fungus, not a virus though. And there's a certain variety called UG99. UG99 was discovered in Uganda in 1999. And there are very few strains of wheat that are currently resistant to UG99. And UG99 has made its way up and down Africa, into Yemen, and into Iran, and is knocking on the door of the breadbasket of Asia. And right now we don't have any good resistance for it. And that's where they need it the most, that's right. So this is what it looks like. This is what wheat rust looks like. So let me tell you a little bit about wheat domestication. We covered this last week. Bread wheat was originated from, or domesticated from, a wild ancestor. I gave you an example of einkorn and durum. Bread wheat's a little different than durum wheat, but the concept is still the same. So the question is, in early domestication or pre-domestication, back at the wild plant over here, was there a genetic bit of information that might have actually protected from this variety of wheat rust that we lost as we went through domestication? As we narrowed the specific set of genetic variation that we wanted, did we potentially lose that? So if we go back and we sequence the wild ancestors of wheat, would we find genes that might give us resistance to this variety of wheat rust? And the answer is that yes, there actually is a gene called SR35. It's one of a number of SR genes that provide resistance to different pests. that seems to provide resistance to UG99. Now, there are lots of technologies that are being looked at right now to try to create resistant varieties of wheat. This is only one of them, one of many of them. But the question now becomes, is there a way for me to cross that specific variety, that specific gene, back in to my domesticated wheat? Is there a way for me to do that? So what I want you to get a sense of is that knowing the genetics opens the door, it lifts the cover off of having to only be able to look at a plant's characteristics that you physically can see. You can look under the hood at the specific genetic changes that are responsible, and you can find those, and you can find those relatively quickly. One example is finding a gene called GMCHX1 that gives soybeans the ability to grow under salty soil, because it this gene actively transports salt out of the cell. Many varieties of soybean don't do well in salty soil. They have a mutated version of this gene. The varieties of soybean that do well in salty soil have a fully functioning copy. If I know that and I have a test that lets me identify that functioning copy, I can follow through my breeding technology and look to make sure that specific fully functioning copy always shows up in the next generation as I'm doing my crosses. Same concept with light-loving tomatoes. I don't know if you know this, because in the South we grow our tomatoes out in the field, but there are lots of places where tomatoes are grown indoors in greenhouses. And they're grown using artificial light. And unlike many varieties where you can use artificial light 24 hours a day, tomatoes can only take light about 16 hours. If they take more light than that, there's leaf damage that actually destroys the plant. So commercial varieties of tomatoes have 16 hours of light and 8 hours of dark. Wild tomatoes can grow 24 hours in the light. So they've identified the specific gene, CAB13. CAB13 is involved in the process of photosynthesis. It helps capture light. And in the commercial varieties of tomato, the specific change in this gene means that after about 16 hours, it's tapped out. It can't continue to function. But the wild variety functions just fine 24/7. So if I can take the wild variety and cross the wild variety into the wild version of CAB13, and cross it into commercial tomatoes, which they actually have done in one strain, the tomatoes grow 24 hours in light, and there's a 20% increase in yield. 20%. That is a major increase if you're a commercial tomato farmer. Now that has to be proven in other strains, but that's the power of this technology. This was the traditional approach, looking at the visual traits. This was what I told you was what was happening at the genetic level, but we didn't have a way to be able to say, hey, do I have that specific gene? Now that we've sequenced the genomes, and now that we can compare different varieties and look for the genetic changes, we now have a marker, a way to identify a marker, a way to identify a specific allele that we want, and a way to create a quick test that lets me, while I have little plants, while I have sprouts, take a little piece of the DNA, a little piece of the plant, get its DNA, and look at a whole flat of seedlings and be able to say, that one, and that one have the genetic marker that I want that gives me the trait that I'm looking for. Those are the three that I'm going to invest my resources in to take to the next level of my breeding scheme. Instead of having to grow all of them up and wait for the trait to appear, which if it's a trait that doesn't show up until I'm at full maturity, that can take a really long time. So that concept is called marker-assisted selection. I just walked us through that whole slide. Sorry, I should have actually gone through it step by step. But essentially, here's the key. I'm using testing of the plants because I know a specific change associated with the trait that I want. I know this version of the CAB13 gene gives me a tomato that grows 24 hours. I know this version of the GMCHX1 gene gives me soybeans that grow under salty conditions. I know this version of this gene gives me a plant that is pest resistant. And if I know that, then I now have a quick way to track which plants have that allele, that specific version I'm interested in, and which plants don't. One more story. I love this story. We do a lot of corn growing in northern Alabama, especially in Madison County. My dad came from a farm. I spent a little bit of time on the farm when I was young, but I have no farm smarts at all. I am a city boy. And we had the opportunity to spend some time on one of our area farms, and I'm really grateful that we were able to do that. I got to ride in the combine, which was really, really cool. And I and my team got to ask a lot of really naive questions about corn and about the challenges of growing corn and deciding what varieties of corn you want to grow to get you the best yield for the least amount of input. A little corn anatomy. Corn actually has two flowers, the male flower and the female flower. The male flower are the tassels that are on top. They produce the pollen. The female part comes in the ear and it produces silks. And the silks essentially connect to each of the future kernels on the cob. So you have to get the pollen from the male tassels onto every single silk, and then it has to travel down the silk and fertilize and create the cob, create the kernels on the cob. So when you get an ear of corn and there are kernels missing, the silk that ran from that kernel out of the ear didn't get pollinated. There's a very special process you've got to go through, and the pollen only is produced under certain good weather conditions. If it's too hot, if it's too humid, that delays. If it's too dry, that sounds different from too humid, sorry, but if there are certain conditions, the pollen will be delayed. And if the pollen is delayed, then you miss the window to fertilize the silks to get the kernels. So if you look at water usage, corn needs a lot of water. But corn needs water at very specific times. So when you reach the stage where there are 12 leaves on the corn plant, your water usage really increases, and it peaks about the time the tassels come out and about the time the silks come out. You need a lot of water in this window, and if you have drought in this window, then you have potentially altered your timing of your appearance of the pollen and the fertilization, and your corn crop is going to be in real trouble. You need less water as the grain, the kernels are what's called filling. So you end up not with shriveled kernels, but with full plump kernels. You still need water, but you need less water in that window. So when you have drought, the timing of the drought is really critical depending on what's happening in this process of tassel and silking and grain kernel filling. So there are a lot of studies, a lot of groups right now trying to develop varieties of corn that are drought tolerant, that will grow well when there's not a lot of water. One of the challenges historically is that you can produce a corn plant that gives you better yield than your traditional high-performing varieties when there's little water, but when there's a normal rainfall, those plants that do well in the drought don't do nearly as well when there's plentiful rain. So that's called yield drag. And you don't want to plant your crop with all drought-resistant corn and then have a full rainy season and you've lost yield. By the flip side, you don't want to plant your crop with all regular corn and then have drought and you've lost out. So it's a challenge, and it's not just if the entire field is dry. There are parts of a field that get less water than others. Now, if you irrigate your fields, that's a totally different issue because there's an increased cost with irrigation. So you're putting more money to get the crop out, kind of as an insurance policy, but you know, there's a higher cost there. So these new varieties of crops that are being developed, they're trying to get rid of drag yield so that they grow well whether it's rainy. They grow as well as others when there's a normal amount of rainfall, and when there's drought, they do better than the regular varieties. So they're using genetic information. So in 2009, the corn genome was published. So there's our reference book. So there are a couple different ways that scientists and breeders are approaching this. They start by working with plants that naturally show tolerance to drought. And they say, what is it about these plants that seems to let them grow well when it's dry? So they sequence the DNA, the genome of those plants, compare it to the reference corn genome, and look for changes to identify candidate genes and specifically alleles, the variety of those genes that might be involved in water use. Another approach is to start with what we know about the corn genome. Here's a gene, this gene seems to be doing this function. This gene, whoops, seems to be doing a different function. And starting with that and saying, what are the genes that I think might be involved in water usage? What are the genes that control when the pores on the leaf open up to let water transpire out of the leaf? And can I find those genes and then find versions of those genes that don't open those pores up quite so frequently or open those pores up quite so wide? Can I identify the genes that control how water is taken up from the soil into the roots? And can I find versions of those genes, alleles of those genes, that maximize that water draw up from the soil? And then you take all that information and you use marker-assisted selection, where I now know I'm looking for this version of this gene and I'm tracking it through my breeding scheme. And I'm going to build a version, I'm going to breed a version of corn that has all the right alleles and all the right genes that make them water sippers, that minimize their use of water, so that when water is not present, they're okay, but they're not affected, it doesn't hurt them when water is plentiful. There are a whole number of groups that are doing this. This is not genetic modification. This is traditional breeding informed by genetics. That is really important that you hear what I'm saying. We will talk about genetically modified versions of corn that are drought tolerant. At this moment, I'm talking about using the power of genomics and traditional breeding schemes, marker-assisted selection. Lots of varieties that are out there. I am not endorsing one variety over another. I'm just going to tell you about a specific variety. And I'm not saying this is what you need to be planting. Nor am I paid any consultation fees or anything by any of these groups. So this is an example. It's called Agricultural Artesian corn, and it's produced by Syngenta. And they have done this process where they found 13 genes that they think are involved in water usage, and they found versions, alleles of these genes that perform better under drought, and they have gathered all those together. They've stacked them, essentially. So they've created this variety of corn, maximize yields when it rains, so there's no yield drag, and increase yields when it doesn't. And so there's all kinds of information on their website about their crop. One way that plants sometimes deal with drought is they roll their leaves. They roll up the leaf. Well, when you roll up the leaf, there's less surface area available to capture the sun to carry out photosynthesis. So you're making less food that then feeds the rest of your plant. So while this is a drought defense mechanism, it doesn't help your crop grow. So the Agricultural Artesian corn doesn't roll up its leaves. These are every other row planted with Agricultural Artesian and the other elite hybrid that doesn't use this technology, and you can see the greener, more green from the Agricultural Artesian. And you can see differences, these are their pictures, not mine, differences in the size of the cob. So it's more filling, more grain, more kernels are produced. Now there are other companies that do this, and you can go to their websites and you can see their technology as well. But this is combining what we know about genetics and what we know about how corn grows. These are their yields. Generally, the further down to the right we go, the less water. So high yields when there's enough water, lower yields when there's not. The gray bars that you probably can barely see, so look to your handout, are the traditional commercially available varieties. The green bar is this specific brand of corn. High or equal amount of yields when you have plentiful moisture, and then when you're in drought, especially in extreme drought, there's a pretty substantial increase. That difference, that on average 15% increase in yields if you buy their corn, may make the difference between if a farmer's crop succeeds or if they lose money. So they have this great ad. We went to the future. We brought back some corn. Now, I found this ad in a farmer's magazine. This was from a July issue. And I started wondering how many other versions of these kinds of ads that talk now about genetics are there. So I looked at 4 issues of this magazine, June, July, August, and September. I found this one. I found this one. We're not reinventing the soybean. Oh wait, yes we are. And I don't think you can read this, and I can barely read this, but this says, utilizing high-performing smart genetics. Smart genetics, but essentially it means we're looking for the specific varieties of our DNA, of the genes that make this, that give us the yield that we want. Here's another one. You're going to want a piece of this. And this one talks about this seed corn is a game changer, engineered with genetic muscle to challenge the status quo. Genetic muscle. I love this. You know, the scientist in me just kind of twitches when I see that. But they're using genetically informed information, marker-assisted selection, to drive their breeding scheme. And then this one. Developed with the genetic code to match your zip code. Now, you hear us talk about personalized medicine for healthcare all the time. This apparently is personalized agriculture based on your zip code, but it's the same concept. There will be multiple varieties of corn plants specific to, I have a region that has a high set of nematodes. I have another part of my farm that is marshy, another part that's dry, another part that has this, another part that has that. My climates do these sorts of things. So here are the varieties. We've always had lots of varieties of corn that perform well under other conditions, certain conditions. We now are using what we know about genetics, and not just for corn, but for wheat and for barley and for all different kinds of fruits and vegetables. It really is a game changer. Before I let you go, the director of the Jackson Center, Daniel, handed me this today. He said, this came in my inbox from a vegetable producer. Colette's. This is a brand new vegetable. This vegetable is a cross between kale and Brussels sprouts. Yeah, some of you are like, ugh. It is not genetically modified. It is a specific breeding scheme. Some of these plants, because kale and Brussels sprouts share a distant genetic relative, you can crossbreed them, and you get something that creates a kale-type vegetable that was easy to prepare and looked great. They can be sautéed, roasted, grilled, or eaten raw, and they have a nutty flavor. A fusion of sweet and nutty. So these kinds of things, again, this is not genetically modified. This is not even induced mutation. This is breeding, standard breeding. Is this natural? This is a question that you've got to answer. It doesn't use any abnormal artificial techniques, but it certainly is something that we didn't have before. So I hope what you take away from tonight, and I've kept you long, so I apologize. I hope what you've taken away from tonight, number one, is that the field of genomics is transforming crops, because we now finally have insight into why crops grow and perform the way they do, why the fruit looks the way it does, why it tastes the way it does, what its nutritional value is. And this is a huge changer for how we look at crop breeding and being able to use genetic testing to follow or track the specific change that we want as we build a new breed. Again, we didn't talk about anything at all tonight that was genetically modified. This is all done. Now, this induced mutation where you're zapping it with radiation or chemicals, again, that's not genetically modified, and our world has said that that is completely acceptable and hasn't said that that needs to be labeled and we can make it organic. So this concept of nature, what is natural, I want you to realize it is a very fluid definition. Now, next week, we're going to talk about livestock improvement. So we're going to transfer the same kind of stuff we've talked about now with crops to beef and chicken and pork and maybe some other things. We'll talk about traditional breeding. We'll talk about how genetic testing, genetic markers are currently used in livestock to assess which parents we should make our breeding stock from. And then we're going to talk about some of the newer technologies in marker-assisted selection. So thanks for spending tonight with me. Those of you at home, thank you for watching, and I'll see you back next week. Have a good night, everybody.